Well, thank you everyone for joining us for today's program. It's part of our series called D3D, Data, Drugs, and Diagnostics on Advances in Tissue Imaging. My name is Joanne Geer, and I'm the Executive Director of the Biopharma Research Council. We are an association for scientists across all the many silos of biomedical research. So that includes industry, academia, nonprofit, government, and supplier labs, and their teams. We run educational programs throughout the year. We do a number of webinars such as these, and conferences ranging from a CRISPR conference we did this spring in Boston, a cybersecurity and pharma event that we did this summer in Princeton, and we have a few things coming up. Just last week we did a program on altering the microbiome at the National Cancer Institute at Fort Detrick in Frederick, Maryland. Of course, today's program on advances in tissue imaging. And in a few weeks, we're going to North Carolina for the Triangle Biotech Research Symposium, which is focusing on convergence this year. We have a number of other webinars coming up that you'll be hearing about later in this series, and we really welcome your participation. Most of the programs that we do come from ideas from our community of about 9,000 researchers internationally. So we're always very happy to hear from you, any feedback, thoughts, ideas about ways that we can work together, and so on. I'll be turning it over to Dr. Matthew Pettijohn in a moment to introduce our topic and to introduce Professor Christian Schwertner. And I'd like to let you know that you're invited to introduce your questions at any time. We're going to take the questions all at the end, so if it's about a particular slide, it's very helpful if you know which slide it was, or a little bit of identification. And yes, the recording and the slides will be posted on our website, and we'll be sending out an email to let everyone know when they're available, so you can refer back to them at any time. And now I'm going to turn it over to Dr. Pettijohn.
All right.
Thank you so much, Joanne. My name is Matt Pettijohn. Again, thank you to you, Joanne, and the Biopharma Research Council for working on our projects. What I'd like to do right now is introduce in a few slides why we are interested in non-destructive tissue imaging, and we'll do a deep dive today on histoindexogenesis non-linear microscope. And our next webinar in October will focus on the light CT, which is a full-field OCT system. We are extremely interested in these non-destructive tissue imaging platforms because they have the potential to eliminate the growth room in the pathology suite and enable direct biopsy to image diagnostics. We are not there yet. What we can do for now is biopsy to image quantification on
Excuse me, Matt, you're going in and out sound-wise.
In tissues of 100 to 300 micron thick in 2D and 3D. Is it, I am on a good line. Is it better now?
It's better, yes. Thank you.
Right. It happens. We had a little bit of a web fluctuation here. So I was reading a few bullets here on these slides. These are very interesting techniques. They are very promising for clinical applications. Right now, we are developing their applications as research-only use. For academia, but also in the drug discovery space, as they have the potential to be fully translational. So when you look at pathology and tissue handling, there are basically two major processes. The frozen section is a quick process at point of care, surgery place. It's very useful for decision support, triage, structure is key on the frozen section, and there is the more complex H&E process that has full diagnostic capabilities. The two systems that we are talking about on these two webinars have potentially their place on both workflows. Light CT is a system that is extremely friendly to use and can be located at point of biopsy. The Genesis as well is friendly, but a little bit more complex in its utilization, as it's sensitive to chemistry, and interpretation might be a little bit more complex, but more powerful at the same time. Everybody will ask us, how does those systems compare to H&E, and we'll address that in detail. Today's webinar is about Genesis, and I would like to turn it now to Professor Christian Schwarzner. Christian is joining us from Germany, and he's professor of urology and urological oncology in Tübingen. Christian, thank you so much for joining us today, and we're going to turn the presentation to you. You will show us what you are currently doing from a research perspective on the Genesis system.
Well, thank you very much. It's really my pleasure to be with you guys. Even being on the other end of the Atlantic, I hope to be able to give you a short overview of what we have been doing so far and what we have gathered up to this point with our Singaporean colleagues. As you have seen, and as Matt has said before, I'm a prostate cancer guy, so I'm a uro-oncologist and by right a surgeon. But we also look a little bit into research, and that's why we came across that sort of a new imaging modality for histology that we find very interesting. So let's go ahead. So my slide's not moving. So the whole plan of this presentation is to just give you a brief overview about prostate cancer, then just a short overview of the stain-free 3D imaging technique. Our idea of whole mount sections that we have used for validation purposes, biopsy Gleason grading, which is in part provided by our Singaporean colleagues, some results on that, and ultimately closing it down with some conclusions. The problem with prostate cancer is that prostate cancer is obviously the most frequent cancer in males, so it is a significant problem to the society, not only in terms of healthcare, but it's a social problem as well. Histology is usually graded according to Gleason, being the standardized reporting system being done by a uropathologist. Screening is done on a PSA-based fashion, and your diagnosis is actually done after a transrectal ultrasound-guided biopsy. And one of the problems with prostate cancer diagnosis and treatment is that there is a significant amount of overdiagnosis and overtreatment because we tend to treat low-risk cancers in a too aggressive way, whereas some of the more aggressive cancer might not be treated appropriately. So we believe that there is an urgent need for better decision-making tools to give us a better risk stratification. Up to now, we are doing a simplified risk stratification based on PSA, so serological testing, the Gleason score, and the clinical T-stage, leading to the so-called D'Amico classification, which is classifying your cancer into a low, intermediate, or high-risk group. And the therapeutic options are actually according to the profile. So if your cancer is a low-risk cancer, you have more options as compared to a high-risk situation. So just briefly, there are many options in the low-risk population, with active surveillance being the most important one. So basically leaving your anatomy unchanged and you won't have any symptoms. On the other hand, you have more aggressive treatment like radical prostatectomy and radiotherapy plus or minus anti-hormonal treatment, so a much more aggressive treatment. The problem with histopathology in prostate cancer is that the core quality is very variable, and we have a significant amount of understaging and undergrading on biopsy. So some of the cancers are actually more aggressive when you look at the ultimate prostatectomy specimen. On the other hand, we are also, as urologists and surgeons, we are also interested in radical prostatectomy and point-of-care assessment of the margin status. So we don't want to wait for the pathologist to come back with the results, but we'd like to have some information on the periprostatic an extension also already in the operating theater. So this would be very interesting for us. And as I said, there is an urgent need for standardized automated reporting for prostate cancer pathologies, because there is a significant inter-observer variability between pathologists, especially when distinguishing between low and intermediate risk cancers, leading to a false treatment. The system that we've been using, you've already seen that slide before in Matt's slides, is the Genesis system that is made by HistoIndex, a Singaporean company. And they're actually the first ones to provide a stain-free 3D quantitative imaging for the visualization and staging of fibrosis, as well as other connective tissue alterations, not only in cancer but also in other fibrotic disease states. You may use frozen, paraffin-embedded, and fresh tissue at the same time, so you're not restricted to a single tissue processing method. The way it works, it uses basically two different image processing parts, one being the two-photon excitation fluorescence, and the other one being the second harmonic generation. I'll come back to that a little bit later. The big advantage is that you can use unstained tissue, which is totally unaffected by any dyeing or photo bleaching, so your slides can be kept for a long time, and there is no photo bleaching, just like in H&E, and you can look at your specimen in a 3D fashion, giving you a lot more insight into the actual anatomy as compared to a standardized histology reporting. As I said, the Genesis is consisting of these two different imaging softwares or ways of imaging. Please excuse my expression, since I'm not a pathologist by right, so I really had to understand that myself very, very slowly. But one of the two, the two-photon excited fluorescence, is meant for cellular morphology, whereas the second harmonic generation is mainly looking at extracellular matrix, and the two are actually merged and then give you an image of both cellular anatomy and extracellular matrix at the same time. And as I said, all this is being done without any staining. This is just briefly showing how it works and how the two systems are actually being conjoined at the end of the procedure. So it's a white light source with a standardized camera, and then the two images, so the one for cellular anatomy and the other one for extracellular matrix, are then merged to one overlapping image, giving you a complete information about the tissue that you're dealing with, and this is all being done by laser excitation. Trying to go further. Again, this is a machine where you can put your slides into this little little ditch, basically, and the system then analyzes the anatomy more or less automatically. So what we have done, we were first interested a little bit more in radical prostatectomy specimens. So we looked at whole mount sections of radical prostates in order to try to assess the margin status, and in order to try to validate the capabilities of this new tissue imaging system as compared to standard histology. I talked about this a little bit before. So when we are doing radical prostatectomy for more advanced or more high-risk prostate cancer, we are very much interested in preserving the structures. This is the prostate, in preserving the adjacent structures that are relevant to minimize morbidity. So both to preserve erectile function as well as continence. So it's very important to have good intraoperative guidance. And some of this intraoperative guidance we already have beforehand from accurate MRI imaging, so we pretty well know where the tumor might be located, but this is just something like an approximative method, and there is still some room for the human error, so to say. So what we did in our pilot study, we looked at the radical prostatectomy whole mount sections, that all have actually had standard H&E assessment. They were digitalized, and the cancer foci were graded according to the Gleason score and marked accordingly. After that, every other section which remained unstained was analyzed using the Genesis system, and the two were actually compared to each other. And then these are the initial results that we obtained. One is that the prostatic microanatomy, this is just a whole mount section of a prostate. This is the urethra, this is the peripheral zone, and this is more or less the central zone of the prostate. And all the microanatomy of the prostate could be nicely visualized, and even a non-pathologist could nicely understand the different subsections of the prostatic anatomy. The prostatic glands are actually dispersed in the fibromuscular stroma. This can also be seen quite nicely, and the stroma generates a pretty strong signal due to the abundance of collagen and connective tissue, whereas the acini were quite strongly autofluorescent, being located inside this fibromuscular stroma. The prostate typically also has some benign hyperplasia, which is usually found in the transitional zone, so around the urethra. And this BPH, which is a benign sort of neoplastic change, could be nicely seen, and it was well demarcated from the fibromuscular stroma that was basically encaging the glandular structure. So this is the autofluorescent, and this is the same image using H&E. Then when it comes to cancer, first looking at Gleason 6, which is a low-risk prostate cancer, still maintaining some of the typical prostatic anatomy. So you can still see the glandular anatomy there, and there is some stroma in between, and this could also be quite nicely seen using the Genesis system. When it comes to differentiating between low risk, so Gleason 6, and high risk prostate cancer, the same technology was also able to delineate Gleason 6 and Gleason 8 quite nicely. So this is again the same slide. So this is high risk prostate cancer, so densely packed epithelial cells, no lumen, whereas in the low risk part of the tumor there was still a lumen. And this could also be quite nicely seen on the non-stained images. So this could be quite nicely validated. So in conclusion, when we look at the whole mount section, I think non-stained imaging gave us quite a good image of the actual prostate cancer. In the next step, and this was mainly done by the Singaporean colleagues, we tried to look at prostate biopsies, also in an attempt to provide something like a point-of-care test for prostate biopsies. So in order to tell your patient straight away you have cancer or there is no cancer. So these are two typical prostate biopsy cores. This is a positive sample, including prostate cancer foci, whereas this is a negative sample, so there are no prostate cancer areas inside this biopsy. Again, this is non-cancer, which can be quite nicely seen, and this would be a non-cancerous region in a positive sample. So a positive sample is not always consisting of cancer only, but it usually also contains some normal tissue that is free of cancer. Whereas if you look here, this would be a cancerous region harboring prostate cancer. In an attempt to try and standardize the image analysis, we randomly took patches within these cancer areas. And as you can see, there are a number of patches that were really taken randomly, and these areas and these patches were then subsequently analyzed using the image analyzer and a collagen channel for a normal sample. And as you can see, these are all the patches that were marked before, and they also, all of them generate a typical spectrum, and they are then basically superimposed. And the idea is to have a typical signal for benign prostate as opposed to a typical signal for low-risk prostate cancer, and a typical signal eventually for high-risk prostate cancer within the same patient. In the next step, we again look at collagen channel at the same analysis for a prostate biopsy core, comparing cancer to non-cancer within the very same patient, and again randomly taken patches in the respective areas. And as you can see, this is the typical spectrum of non-cancer within a biopsy harboring prostate cancer. Again, a typical spectrum that is being generated, and when this is compared to the cancer part, you can nicely see that this is giving you a totally different spectrum. So there's a lot more collagen, a lot more stroma being found in the cancer as compared to the non-cancerous area. And this whole background is actually meant for standardized reporting at the end of the day. Again, these are the same analysis, so normal sample compared to 3+3 cancer, and this is ultimately then compared to a 4+3 cancer. So this would be a cancer that requires treatment, whereas this would be a cancer that would be best handled by active surveillance, because the mortality of low-risk prostate cancer is extremely low, and usually patients die much faster than the cancer would actually grow. Whereas here, these cancers can be lethal in the long run. So as you can see here quite nicely, there is a distinct pattern for the two cancer subtypes, with the more aggressive one having a more heterogeneous pattern. So just heading to the next one. These features can be visualized in detail, and this is just to show you that there are different things that can be measured within such a panel, so including the distance between the collagen fibers, the standard deviation, and all that kind of information can be taken out of each panel, and then ultimately all this can be assessed statistically in order to give you a good basis for standardized reporting. All this can also be done in a different imaging, or let's put it different, in a different visualization platform. So this again is a 3D feature space showing again 3+3 cancer, 3+3 cores, areas with no cancer, and again 4+3 cores and non-cancerous sample. And if you look at this image here, this looks a bit messy, but once you sort of filter for the cancerous areas, you would get a much nicer picture, and you're quite easily able to distinguish between the 4+3 and the 3+3, which really has a significant impact on treatment of your patient in the long run, and it probably helps us to reduce morbidity in the long run. Again, this is clinical data from the hospital, basically straight out. Again, a biopsy core, and again looking at different areas, cancer versus non-cancer, and we try to apply this algorithm that I showed you before. And again, this algorithm actually is quite nicely able to distinguish between Gleason 6, a low risk, and intermediate risk prostate cancer. So obviously we are in the middle of validating our findings now, and we hope that this pattern that we have detected up to now can be refined in the long run. This is a different way of showing you, and the whole meaning of this slide is I won't go into every single picture here, but it just shows you that you can nicely differentiate between intermediate risk and low risk prostate cancer on all the different patches. So every single image here means a different, is actually reflecting a different patch and giving you a nice differentiation between the two cancer subtypes. So in conclusion, non-stained imaging can actually provide good anatomical details in the prostate. BPH and inflammation as well as cancer can be clearly distinguished, and we hope that this, or we still believe that this might be an ideal tool also for intraoperative decision making in the operating room, as soon as there is standardized reporting available, and we have shown that standardized reporting might be feasible. There is still some homework to be done, so we still need to correlate our findings with long-term clinical outcome in order to see whether the findings we have obtained in our study here are really relevant in the long term clinically. In the future, we hope that non-stained imaging might enter the operating room as a point of care test system to optimize our operative results and also to give quicker information to our patients in the biopsy setting. Thank you very much. This is a joint collaboration between HistoIndex in Singapore, Singapore General Hospital, and our institution in southern Germany. So this is southern Germany. And we have the very same weather right now. Thank you.
Thank you so much, Christian. Joanne, I am going to continue to talk about this technology and how it can be applied to research purposes. If you kindly give me control so I can share my slides.
It should be working now.
Very well. Excellent. So Christian has done a terrific job to show you the potential of this novel imaging technique in the clinic, and as he said, there is still homework to do to make sure that this is a diagnostic level technology, but you have seen that both the imaging and the data processing algorithms that are being developed and studied have the potential to get there. The purpose of my presentation is to explain you what we're doing here in Princeton to make this technology available for all of you here in the Northeast. And actually, we are currently working with California as well. This would be studies that we can do in research, both in pharmaceutical companies or with academic centers. We are opening, and we have opened the application lab in Princeton, and we're offering technology access options. The Genesis is a 400K system all combined. So it can be an expensive program. It has full production capabilities. Christian could have mentioned that we can do 10 slides per batch and up to 10 protocols per slides. But we believe that a nice technology access option would be to offer services. Let me bring you up to speed on what is our focus right now, because of course we want to make sure that the services that we offer and the application that we offer are well understood by the staff. And we have decided to focus on fibrosis, mostly because fibrosis runs across several therapeutic areas, and we are mimicking what several pharmaceutical companies have done, is really to create transfunctional fibrosis study groups that really might start in liver and then expand in lung, heart, kidney, skin, or elsewhere. So everything we learn and we share on fibrosis has translational opportunities both across therapeutic areas and, as we understood from Christian, all along the development of a product up to the clinic. One of the things I'd like to stress, and Christian explained a little bit how the system works, we indeed have two optical methods. These optical methods can be tuned to the fluorescent signal that we get from the proteins inside the cells, but also to the signals we get from a second harmonic generation perspective. The tunability of the system here helps us activate different proteins in the cells or different second harmonic generation signals. So from a research perspective, we can really look at a wide range of fluorescence signals, which we will call at some points biomarkers. These are stain-free fluorescence signals, but as well from a second harmonic generation, we can look at different signals from collagen or other extracellular matrix signals. So keep in mind that the systems might be complex, but it's applicable to many diseases, and we can also do deep dives into different biomarkers, if I may say, coming from the two-photon fluorescence aspects. In addition to the image capabilities that the system offers, Christian stressed the fact that image analysis quantification is important, and we have developed in collaborations with Singapore and validated quantification techniques that are very relevant to collagens and fibrosis. In terms of collagen and collagen strings, we might quantify too much. Here is an overview of all the collagen strings parameters that can be measured. Let me remind you that we work as well in 3D, so these things can be measured in 3D as well. And when it comes to the red signals is NADPH in the cells. We are able to quantify as well at every cell how much NADPH signal we get, but also when NADPH changes in quality, which can be related to the cell damage, we observe a shift in the wavelength of the emission, and that shift is a very precious information that can relate to the damage that we have in the cells. So the technique can be extremely, extremely sensitive, and this is why we believe it has a huge potential when we do research. In terms of quantification, this is an example of the collagen quantification techniques that we can use, both in terms of, for instance, measuring the aggregated collagen and the distributed collagen. And this is a very conventional CCL4 fibrosis model for the mouse, where we see an increase of total collagen according to the research protocol. And when we look into details, we would see that the aggregated collagen goes up, but the distributed collagen goes down. Second harmonic generation can also separate collagen 1 from collagen 3, and that could be a very, very powerful measure in your research programs. When it comes to cell damage quantification here, we look at NADPH two-photon fluorescence and are able to quantify the amount of fluorescence that we receive from the tissue and stratify this amount of fluorescence against the protocol that you use in your research. So I'm going to call that basic quantification. We have published more advanced quantification techniques, and those become organ-specific. This is, let me give you a quick overview of the Q fibrosis. The Q fibrosis incorporates both the collagen information and the NADPH information that are weighted, filtered into one single index. So we are taking those two signals, we are treating them, and we are ending up with one single parameter that has been proven to be extremely reproducible and very, very comparable to the METAVIR score that is used in clinical applications. So as you can see here, we have the Q fibrosis score, the METAVIR score. There is a good correlation and a good separation of those values. So although we are not ready to apply that in the clinic, I think it shows the potential that this technique, imaging technique, combined with sophisticated image and quantification techniques, can really help you stratify and really look at very suitable outcomes in your fibrosis-related program. I'm going to breeze through a few slides. The slides will be available after the webinar. I'm just going to show you very quickly different organs where we have looked at fibrosis. Liver disease is an area where we are very, very strong. We can see inflammation areas, fat as well for this non-alcoholic fatty liver disease. Kidney fibrosis is also in our reach. This is a full slide image where we can definitely look into different levels of fibrosis. We can do heart as well and myocardial infarction, where we can image different tissues. As a matter of fact, today we are imaging heart sample that we received from California, and lung cancer as well. I'll be happy to answer questions at the end if you have specific questions on those different kinds of fibrosis. Let me tell you about the workflow that we use. It's basically a on-demand workflow. We have two systems in Princeton, and typically what we would do is to have a discussion. Our scientists would have a discussions with your teams and yourselves to really understand what is the scientific question behind your protocol. It might be to create a baseline, it might be to investigate a specific outcome. We would then obtain a PO from your teams and receive the samples. We receive them directly mounted, although remember, we can do fresh tissue imaging on the system. Once we receive the samples, we are applying the protocols, the imaging protocols that we have discussed. We process them by batch. As I said, we can do 10 samples, 10 slides in one batch, and up to 10 protocols on each slide. Full slide imaging takes time, so we like to be very focused and look at specific fields of use that we can definitely define together. And we basically then move to the image processing. I'm going to show you how we share the images back with our clients and we do interpretation. But the big idea is that we ship you back the samples. They have not been damaged, and you can continue your usual workflows on those samples. We have implemented a very friendly cloud-based pathology image share workflow. It's a new technology that comes out of University of California, which works like a Google share. We would put those images, and you have access. We can collaborate basically to look at the slide. This is the full slide image and the field of view that has been chosen, and then you would have the full image. So these are big files that can be zoomed in, just like on Google Earth. Of course, this is what we propose. We fully understand that some images would need security, and we can work with your own digital imaging workflows if they are secure. I just wanted to stress that we can collaborate when it comes to defining the best algorithms to interpret, quantify your images. So in short, in blue would be your samples. They come, we image them, we send them back to you, to the next workflow. And while you move on the next workflow, we work on the digital images together and the interpretation together, so you get the most of your research and time with us. Hopefully, these very first experience and services that you will outsource to us will give you the opportunity to generate data, to really understand the power of those technologies, and hopefully work across your therapeutic areas to build some plans to access the system. Thank you so much for your attention. I hope we gave you a broad perspective of what Genesis can do, both in the clinic and in a research environment. We would be very happy to take your questions.
Well, thank you, Dr. Petitjean. This was a terrific program. We have some questions from our community, so I will put them out, and either you or Christian are welcome to address them. Let's see. How is the application of this non-destructive tissue imaging technology used in the drug development for prostate cancer patients?
Well, you want me to? I can try and address that. I think that when dealing with prostate cancer drugs, you're usually dealing with more advanced cancers, so namely metastatic prostate cancer. And in olden times, we just treated the patients with anti-hormonal drugs, but now we are taking more and more biopsies out of the metastatic lesions in order to understand the individual changes that the prostate cancer has undergone over time. So we think that by taking biopsies and looking at them using non-stained imaging might be something like a biomarker for certain drugs. So changes in the extracellular matrix might be something like a surrogate parameter for certain cytokines that are being expressed in the lesion, and might also be used as a marker for, let's say, small molecule drugs. So I think there is quite a lot of potential to it.
Thank you. Did you want to add something, Dr. Pettersson?
Yes, I think the question can be also split, whereas we're looking at clinical research or preclinical research. I think Christian gave a good example of what can be done in clinical research. In preclinical, we would definitely establish baseline on existing models. So you can definitely, for instance, build a baseline with us, whereas we receive the slides. We image and agree to quantify specific outcomes, and then you continue with conventional techniques, and then you create your correlation aspects. Once you have done this calibration baseline correlation aspect, then you might want to explore how the animal responds to your treatments. And obviously the baselining and the validation is important to be done. It's some work to be done, but we believe that the sensitivity of this technique and the quality of the outcomes, the index that we produce, can be very relevant in early stage of drug discovery.
Thank you. You may have sort of begun to answer this next question, but I think it puts the point well. In your view, how is this non-destructive tissue imaging technology used for precision medicine and treatment?
Am I supposed to answer the question?
Yeah, Christian, I think that's a good question for you.
Well, it's actually pretty much the same thing as I just said. I mean, we are not only looking at prostate cancer like in a metastatic state, but we'll also try and understand localized prostate cancer a lot more deeply. And so if we have the chance to use this new modality to really isolate the non-lethal prostate cancer, then I think this would be the first step towards individualized treatment, especially of the low-risk prostate cancers. And on the other hand, the tool might also be helpful for new treatment options like focal therapy. So when we are trying to see whether focal treatment has been sufficient to really ablate the cancer or the cancerous lesion from the prostate. So I think they'll be offering us a great deal of new information.
Yes, Christian, I totally agree. We are investigating the same things here at NYU. On liver, which is a different organ, on liver, for instance, HistoIndex has created a smaller system that is dedicated to liver imaging. And the end goal of that system is to be in the clinic. And the quantification aspects bring one more index to decide what is the best way forward to treat liver disease. As you know, there are many ways to characterize a liver, and the more indexes you have and the more balanced the more triangulation you do on those index, the better outcome you can propose, or the better treatment and targeted treatment you can propose to your patients. So the story is true as well for liver, and we have data on that.
Thank you. We have a new question. Imaging is a surface analysis tool. How is 3D imaging done? Is that through cryoslicing?
I think, Matt, you're better in answering this question. I mean, I can do this as well. I think it's not cryoslicing, but you basically can look into the specimen. But Matt can probably provide more information on that.
Yes. So let me answer first. It's a surface analysis, absolutely. Let's take a tissue. The light penetrates into the tissue, and we are using in HistoIndex a femtosecond laser. So penetration capability is pretty high, and we can get backscattered photons and transmission photons that, thanks to instrumentation, we can read pretty well. So there is a slicing ability of the technique, and we can do images in a volume of from 0 to 300 microns. And now, when it comes to a bigger sample, for instance, we are running a study on small animal brains to look at potential Alzheimer's disease outcomes. We are working with the pathologists. So they take the full brain, and they can do 4-micron slides or 40-micron slides. We can image the 40 microns in 4 sections, and when we work across the different sections of the brains, we can rebuild the brain. It's a little bit tedious to do this kind of studies. It requires very good pathology and tissue preparation skills. We can do the imaging easily, but that's a way to do full organs 3D images. But it's a little bit tedious when you're looking at more recurrent protocol. I would suggest to look at 40 micron slides of the tissue area of interest and do 4 sections, and that can be done automatically.
Thank you. What are the speakers' thoughts about using these assays in companion diagnostics? That is, are they partnering with any drug company to submit a drug assay complementary filing?
Let me. This is a very good question. That's where we're going. We're not there yet, because as you might know, these projects take time, are expensive, and we need strong partners. Definitely, we are looking at the potential of these stain-free digital pathology techniques to be able to obtain diagnostic level information. From a regulatory standpoint, it's a little bit different from when it's out there on the market, but because we measure protein signals and we also have access to extracellular matrix signal, we believe that we'll be able to bring something very compelling to the diagnostic community, but we are not there yet, and we're looking for partners. So more to come in a year or so.
Oh, lovely. Let's see, another question. Do you think availability of fluorescent markers that never photobleach and can be excited by 2 photon excitations will still be valuable in your approach?
Joanne, can you repeat the beginning of the question?
Sure. Do you think availability of fluorescent markers that never photobleach and can be excited by 2 photon excitations will still be valuable in your approach?
The physics would work. I agree with that. Our mission is to go stain-free, so we are not sure that we would like to embark in projects when we add biomarkers that can be excited by 2-photon fluorescence. Having said that, I totally agree that there are intrinsic, or I should say stain-free, markers in the cells that we can access through 2-photon emission, and those are the ones that we are looking for. We are exciting the tissues at a very specific wavelength. We are collecting fluorescence, and we have different filters to really select the peaks of emissions of interest. And this is an area that we are very excited about because we can look and probe many different proteins inside the cells. So that's the way we're going with the instrument.
I'm going to combine or ask you 2 questions that I think are related. One is, have these assays been used in clinical trials or primarily as research only? And are there any plans to submit reagents to the FDA for marketing approval?
Well, from a urology perspective, this has all been done just for research purposes, so no proper clinical trials yet.
And regarding clinical trials, nothing in the US right now. There are studies outside of the US. Do we have plans for FDA submissions? Yes, we do. I can take this question offline, otherwise it's going to be a little bit boring and technical. But yes, we have some plans to make those systems available. It will take time, and it will require, indeed, clinical studies, depending on the ambition that we might have for the intent of use.
Thank you. I think we've started to address this question, but you may have more to say. How do you envision this quantitative tool as part of integrative diagnostics?
So I'm going to have to admit my ignorance about the terms of integrative diagnostics. I don't really understand what that means, but let me try to answer that. If we're talking about a panel of diagnostic around a specific condition, we believe, for instance, in liver, that the Q fibrosis index that has been developed and validated, at least on a research perspective, brings a point of information that can really enrich the quality of a liver diagnostic decision. So from that perspective, when combined to additional diagnostics such as blood tests or the other liver diagnostics, the Q fibrosis enhance the quality of diagnostics. Now, diagnostics is a very complex science. In order to be valid, it requires very sophisticated data collection, interpretation, and we have not obtained this kind of data right now. But definitely, it's on our roadmap, and we'll most likely start with liver or prostate.
Lovely. Well, we have a few more minutes if we have any additional questions. And I want to thank you both very much. This has been an extremely informative presentation and very hopeful in terms of the capabilities that we're seeing. I think it's a perfect time for us to, if either of you would like to just say a few words in conclusion, and we'll end our program in a few minutes.
Well, I, as a urologist and as a clinician, I strongly believe in point-of-care tests and in standardization of diagnostic tests. So I really think that we should think more about this, and of course we require validation and of course more centers across the globe really participating in validation studies. Thank you.
Thank you.
Yes, thank you, Christian. And on our end here in the US, we are really excited to bring this technology to so many research centers and pharmaceutical companies in the area and across the country. We want to hear from you, so please do not hesitate to reach out to me or my team. We are very curious, and we are happy to do a few projects to calibrate the relevance of this technology to your applications. Thank you so much for joining, and thank you so much for your great questions.
Well, thank you, everyone. Have a wonderful day. You'll be hearing about us. We'll send an email when the recording is available, and you can certainly share it with your colleagues and lots of upcoming programs. Thank you so much. Take care.